friends, you're back with me, Professor Girish Kukreja. Here I am with you to discuss some of the MCQs related to enzyme kinetics. Yes, these may help you to prepare for your regular graduation examinations and also to crack the various entrance examinations for M.Sc, for Botany, Zoology, Microbiology, Biotechnology, life sciences in general. Okay, so without wasting much time, let's go to the today's question. So the today's question, it says the Michaelis-Menten constant, that is Km, right? We all have heard about this particular constant, the Michaelis-Menten constant, that is your Km, right? Which of the following statements is true for the Km, right? We have heard of this Km. So which of these is true for the Km? Okay, let's go one by one. It says A, this value of Km is equal to the value of Vmax. Okay, we'll go one by one. Then B, it says the value of Km is equal to half of Vmax, right? Some of you jump to the conclusion it is right, but let's come to it. Then you have C, that the value of Km, it changes with the concentration of your enzyme, right? And the D1, the value of Km does not change with the value of pH, okay? And I have like options with me, like I have the first option saying that A, B is correct, okay? Second says that only B is correct, right? The third says that A, B, C is correct, okay? The fourth says that A, B, C, D are correct. And the last one, it says that all are incorrect. None of these is right. Okay, let's see one by one. So the first one which you saw, that the value of Km is equal to the value of Vmax. Like we know that when you plot a graph of your enzyme concentration, not enzyme concentration, when you plot a graph of substrate concentration versus the velocity of the reaction, right, you get a hyperbolic curve. And that particular maximum value where the enzyme gets saturated and works at its maximum is called as Vmax. So obviously Vmax is not equal to Km, right? We define Km as the amount of substrate concentration required to reach half of the Vmax. So what I do, I take half of this Vmax, right? And this half of the Vmax I'll plot, extrapolate to this x-axis, and there I have what is called as the Km. So the second statement which says that it is equal to half of Vmax, remember that is also wrong. Your Vmax half value will be on the y-axis, whereas Km is on the x-axis. So second statement which states that Km is half of Vmax, it is wrong. Km is the substrate concentration which you require to reach half of the Vmax, right? The third one which says that this particular value of Km, it changes with the concentration of the enzyme, is also false, right? Because we know that out of these two constants, the Vmax and the Km, Vmax, it depends on the concentration of the enzyme. You change the enzyme concentration, the value of Vmax it changes. But Km is independent. We know that Km measures the affinity of the enzyme towards the substrate. So the love of the enzyme towards the substrate does not change when there is more substrate or when there is less substrate, because you know that the love should be unconditional. So is the love of this enzyme towards the substrate. So the value of Km, it does not change with the concentration of the enzyme. The last one, it says that it does not change with the change in pH. Why not? Though it is unconditional, but then we know that with the changes in pH, the structure of the enzyme would be changed, and therefore its affinity towards your what you call as the substrate will also change. So that also will result in the change in the value of your Km, because the structure will be distorted and obviously the function will also be distorted. So what you got here is all the four options, they were wrong. Yes, so we have the last option here, the fifth one, that all the statements are wrong. So what you have to remember, the crux is that Km is not half of Vmax. Most of the students will do this mistake. Km is the substrate concentration required to reach the half of the Vmax. We'll talk about the affinity and the Km and the relationship in some other videos. And if you would like to know about the derivation of this Michaelis-Menten constant, you will get the link of the video in the description box. So stay tuned with me, Professor Girish Kukreja, for more in life sciences. Thank you.